Good day, doctor. My name is Janet. To answer your question, considering that there is time constraint and an urgent need for the treatment of COVID-19, my idea would be going for the strategy which is repurposing of the existing drugs, which means using the currently available antiviral drugs to treat diseases other than its intended target. And since those drugs are already available on the market, such as acyclovir, ganciclovir, lopinavir, oseltamivir, remdesivir, ritonavir, and others, much information is already known about them. For example, the chemical and physical properties of the drug, the route of administration, bioavailability and pharmacokinetics, how to synthesize them and manufacture them on scale, etc. The drugs have also undergone much of the preclinical, clinical, and toxicity studies. Therefore, knowing these elements can actually help us to go from the early discovery stage to impacting patients a lot quicker. And this can save us time as well as money when in developing the drug for COVID-19. On top of that, repurposing drugs already approved for other diseases are known to be largely safe. This strategy is now being used in the Solidarity clinical trial for COVID-19 treatment, which is sponsored by the World Health Organization, WHO, and its partners. For example, the two HIV drugs, which are lopinavir and ritonavir, are now being tested due to the possible similar action on the coronavirus. In fact, the use of the AZT drug, which is the azidothymidine, during large viral outbreaks some 30 to 40 years ago with HIV, was actually discovered through screening of the cancer drugs. Therefore, I think that doing virtual screening on all the antiviral drugs available currently might actually give us a lead in finding the potential drug for COVID-19. Moreover, since the SARS coronavirus 2 and the SARS coronavirus are genetically very similar, having about more than 90% sequence identity in the essential enzymes, and share similar entry receptor, which is the ACE2. This suggests that the antiviral drug for SARS coronavirus might actually give a possible therapeutic effect to the SARS coronavirus too. Given that there is already a way of characterizing the structure of the virus main protease in in the SARS coronavirus 2, which is the essential enzyme that is responsible for completing its life cycle, we can easily use the docking software to predict how the drug interacts with the receptor or the enzymes. With better understanding on the drug interactions, we can alter and optimize the structure of the drug, the structure of the existing antiviral drugs, to specifically target the site of interest. But then, of course, we need to take into account the variable interactions that happen between them. On the other hand, if the structure of our intended target is unknown, we can still screen the antiviral drugs and identify the pharmacophore. And then we can make changes to the structure of the drug to better increase the interactions while retaining the pharmacophore. A QSAR, quantitative structural activity relationship, may be derived. Meanwhile, we can consider drug combination therapy, which might give beneficial synergistic effect, like in the case of the two HIV drugs. So because lopinavir is quickly broken down by the proteases in our body, therefore it is used together with low levels of ritonavir to actually help the lopinavir to persist longer in our body. Other than that, a group of Chinese scientists have actually successfully decoded the genetic sequences of the SARS coronavirus 2 in January 10th this year. Given the viral genome sequences, we can actually predict the virus history. Like for example, we can know how fast do they actually mutate and where in the genome do the mutations actually happen. And from there, we can keep up with the mutation of the virus by changing the structure of the drugs accordingly. So the viruses, they are obviously smart, so we need to be smarter and think way ahead of them. Eventually, when the new drug is being produced, they still of course need to go through the preclinical and clinical studies to ensure its efficiency and safety. As for the route administration, from the reading I've made, I would suggest to go for nasal spray, the form of a nasal spray, because the virus, the coronavirus, actually infect the cells of our respiratory system. So I think that this approach will be more direct in killing or against the virus. And lastly, of course, in doing so, I would need to hire experts in medicinal chemistry, in bioinformatics, biochemical design, molecular biology, and others. And this is the summary of the key points of that I've made. And these are the list of references that I've referred to. And that's all from me. Thank you very much.